right, well, good morning. I appreciate the opportunity to describe some of the exciting recent progress that have been made in defining the genetic contribution to lupus. So over the next 25 minutes or so, I'm going to begin by describing some of the challenges and the new tools that are now available for defining the genetic contribution for human autoimmune diseases. I'll focus on some recent successes in gene identification, particularly those related to genome-wide association studies. And importantly, these studies have provided valuable clues to etiologic pathways in lupus and related diseases. I'll then describe some of the current efforts to refine genotype-phenotype associations both within and across autoimmune diseases. And then lastly, I'll describe some of the types of studies you can expect to see as we move forward in the GWAS era. So lupus is the prototypic autoimmune disease, and although it's relatively rare, it's related to a broad group of disorders, which together are very common and responsible for a tremendous amount of disability. In spite of the clinical heterogeneity within this group of disorders, though, there are some striking common features, such as female predominance, that suggest some common pathogenic features, including perhaps an important role for genetics. Families such as this pedigree, which I've illustrated here, that's been recruited and enrolled into one of these valuable genetic resources termed the MADGC collection, nicely illustrates how the notion that there may be genes that predispose to multiple autoimmune diseases. What this family nicely illustrates is not only the number of affected individuals by human autoimmune diseases, indicated by the filled-in symbols, but also the diversity of diseases within this single family. I'm not sure if you can see that, but we have an individual with lupus, rheumatoid arthritis, psoriasis, JIA, autoimmune thyroid disease, so a tremendous range of autoimmune diseases within this single family. Now, compared to most other autoimmune diseases, the overall genetic contribution to lupus may be somewhat greater. One common way to measure the overall genetic contribution to a disease is simply by comparing the risk of disease among siblings of an affected individual, to the risk of disease among the background population. Now, this ratio, termed lambda S, S for sibling, provides a measure of the overall genetic contribution to the disease. And as you can see with these lambda S figures that I've shown you here for a range of autoimmune diseases, the genetic contribution to lupus is somewhat greater than these other autoimmune diseases. Now, we know, however, that genes aren't the entire story in determining risk of lupus or other autoimmune diseases, but in fact, most human diseases are genetically complex. That means that there are many, perhaps many genes that predispose to the disease, an important role for non-genetic factors. These diseases don't exhibit straightforward modes of inheritance in families, and there can be a striking lack of correspondence to the genes that one inherits and their risk of developing disease over their lifetime. Furthermore, there is important heterogeneity of genetic associations. We're learning, for example, as you'll see, that individuals with disease represented by different disease manifestations have inherited different genetic risk factors. In spite of these complexities, however, though, as you know, there's been tremendous recent progress in the identification of genes that contribute to lupus and related disorders. I think this timeline is nicely illustrated by this figure from a recent view of this issue by Kathy Moser and colleagues. And in addition to illustrating how much recent progress has been made since the initial identification of HLA region genes became important 4 decades ago, this timeline also nicely illustrates the close relationship between developments in the laboratory and statistical methodology and progress in gene identification. And in particular, as I'll describe now, there's been tremendous recent progress in the last couple of years as it's become possible to perform genome-wide association studies. So these studies have had a tremendous impact on a whole range of human diseases, and I wanted to draw your attention to this nice review of this topic in this recent issue from the New England Journal of Medicine. Now, just in the last 2 years, there have been 4 GWASs completed involving lupus patients and control individuals of European ancestry. We've learned a tremendous amount from these studies. But first, let me describe for you what these studies entail. They're nicely illustrated by this Manhattan plot, so named because of the resemblance, if you're lucky, to the Manhattan skyline. And these studies entail in analyzing and comparing cases and control individuals for hundreds of thousands, if not 1 million genetic markers located across the genome. So in this particular genome-wide association study that involved about 1,300 cases and 3,000 control individuals, it was very clear that the HLA region, STAT4 and IRF5 were unquestionably associated with lupus. We already knew about those 3 genes, but there were some new genes that were identified in this and other studies that came out just within the last couple of years, such as this gene here, BLK, and ITGAM, this integrin. Now, very importantly, however, all of these dots down in this region have very suggestive, although not definitive, evidence of association in this single study. And it has been the follow-up studies that have focused on those regions that have been particularly informative. Now, here's another recent GWAS that was completed, smaller set of patients, which probably explains why there were fewer dots up here in this region, but also a different marker set, and that's probably why, in addition to HLA and IRF5, a different gene was particularly strikingly associated with lupus in this study, TNF-AIP3, and I'm going to talk more about that subsequently. Now, this is a nice example of some of the replication studies, the follow-up work that can really build upon these initial findings in GWAS studies. This study, which was, as is typical now, performed by many investigators at several institutions, led by individuals in Genentech in this case, involved following up the top 2,500 regions from those previous GWAS studies among about 2,000 lupus patients, over 4,000 control individuals. And as the result of this single study, there were 5 new lupus genes identified, listed on this slide, bringing the total to over 20. And there were many other genes that were highly suggested as a result of this study that are being followed up now, in addition to, as I'll emphasize in a minute, a lot of overlap with other autoimmune disease findings. Now, I wanted to show a little bit of the data from this study for these top 5 genes to make a couple of points. First of all, these studies are designed to identify common genetic variants, as indicated here by their common frequency in this population. Now, nonetheless, the statistical evidence is irrefutable, but the effect sizes are relatively modest, so increasing the risk of disease perhaps only by about 25%. Now, importantly and appropriately, these studies are now being expanded to other populations. There were 2 genome-wide association studies recently published from Asian populations. These are the titles from those recent publications, and this work is now being extended to other populations as well. And I'll just mention for a minute here what's interesting about the extension to these other populations. So this is the Manhattan plot from one of the recent Asian GWASs, a relatively small study. Nonetheless, look at all of these genes that are highlighted from this one study. Now, many of these genes you'll recognize from the previous slides from European population studies. However, some, even of the top hits from this scan, are novel genes that, although maybe have a rationale for involvement in lupus based on what we know about those genes, hadn't been identified in the very large studies from European ancestry populations. So once again, we are likely to find new genes contributing differentially in different populations, probably explains some of the phenotypic differences that we appreciate across populations. Now, importantly, though, in addition to the large number of genes that have been identified in these studies, there's a striking degree of clustering within just a small number of biological pathways that really underscore the importance of these pathways in disease pathogenesis. Now, this is a nice figure from a recent review of this topic that highlights the 3 pathways that have been particularly notably highlighted by these studies. They include genes involved in immune complex processing, immune signal transduction, and perhaps the most emphasis has been drawn to the toll-like receptor type 1 interferon pathway. And let me show you a little bit more about that to give you an appreciation for how striking the association is between these recently identified genes in this pathway. This is a complicated figure. It comes from a recent review of this topic that shows in a nicely diagrammatic fashion many of the genes, the proteins they encode, that are relevant to this pathway. And if you look carefully at the slide, you'll see a lot of those gene names that were just flashed by you in the previous slides. Now, what I want to point out, however, is that if we gray out the ones that, as of about 6 months ago, hadn't yet been identified to be significantly associated with lupus, you're still left with at least half of the genes in this pathway having been clear-cut evidence of genetic association with lupus. And I'm sure that in the coming year or two, many more of these grayed-out genes will take on that color as they are demonstrated in future studies. Now, I want to focus on one of the genes, TNF-AIP3, that I've mentioned and is shown here in this figure over here. I think recent work and previous work related to that gene, I think, illustrates several principles that I would like to share with you. First of all, there was an initial indication that this gene, TNF-AIP3, that encodes the A20 protein, was important in autoimmune disease based on some animal model studies that were done by Avril Ma and colleagues at the University of Chicago 10 years ago. They found that mice that were lacking this gene failed to regulate TNF-induced NF-kappa B and cell death responses. They were very sick. They died prematurely, and on autopsy were striking for the amount of inflammatory infiltrates really throughout all of the organs and tissues. Now, one of the very early genome-wide association studies in rheumatoid arthritis implicated this genomic region, although it was a bit unsettling that the genetic regions that were strongly associated with the disease were a bit away from the gene, not right within the gene. Now, nonetheless, that's turned out to be quite a common pattern. And then a year or so later, when whole genome data became available for lupus, we could also demonstrate that TNF-AIP3 is also important for lupus. And in fact, there's a stronger degree of association between variants of this gene and lupus compared to RA. Now, in one of the studies that we led, we were able to demonstrate that not only is there an effect at this gene region, but there are 3 different variants spread over a wide area, each one of which contribute independently to risk of lupus. I've summarized some of the information on this slide here, and what I'd like to point out is although overall modest, still sizable strength of association between these variants and lupus. And of particular interest, one of these 3 variants was a coding SNP, so it altered the amino acid sequence of the protein, suggesting that perhaps this represented a true causal variant. Now, the fact that it altered the amino acid sequence also allowed for Avril and others to lead some functional studies to try to understand the mechanism of association with lupus. And it turns out that this has been a relatively rare opportunity as part of these recently performed genetic studies for reasons that I'll refer to subsequently. Now, what Avril was able to do was to transfect cells with either the normal allele or the risk allele, and then measure NF-kappa B activity after TNF stimulation. He was able to show that cells that had the minor risk allele were less effective at inhibiting NF-kappa B levels, suggesting a decreased anti-inflammatory activity of that risk variant. So some intriguing data from this particular risk variant recently identified for lupus. Now, it turns out that TNF-AIP3 is probably relevant to most, if not all, human autoimmune diseases, suggesting a fundamental role for this gene in human autoimmune disease. And I've listed some of the recent studies that demonstrate significant evidence of association to this gene or this region. However, the pattern of association across these diseases is by no means straightforward. Now, let me walk you through this slide. Take a minute here. Here, this is the genomic region around the TNF-AIP3 gene, which is indicated here. And each one of these little teeny hatches refers to a genetic variant, and the red indicates regions of high linkage disequilibrium, where alleles at these variants are highly associated with each other. Now, notice how far away from this gene the RA association is, as well as a more recently described type 1 diabetes and celiac effect. Now, the lupus-associated variants are within the gene and way over here and way back over here. The Crohn's effect is here, the psoriasis effect is here. So it remains to be seen whether what is going on here is many different causal variants that are differentially related to these diseases, or perhaps there's a common effect that we haven't yet been able to to identify or precisely localize through these studies for various reasons having to do with different markers, et cetera. So this is a pattern that's emerging in spite of the clinical heterogeneity of this group of disorders. To a surprising extent, genes identified in one autoimmune disease we're now finding are relevant to others, as nicely illustrated by this diagram here. Now, most of you are very, very familiar with lupus, of course, and you know that even just considering lupus by itself, there's tremendous clinical heterogeneity encompassed. So let me show you some of the recent work that shows the differential relationship between genes and disease characterized by different manifestations. And I'm going to show you an example related to the STAT4 gene, which was recently shown to be related both to lupus and rheumatoid arthritis risk. And we were interested in determining whether STAT4 is associated equally well with all types of lupus, or whether maybe it's specific to one type of lupus versus another. And we found a striking variation in the strength of association according to the specific manifestations in the patient. So, for example, the frequency of this risk variant, which was about 22% in controls, went all the way up to 38% in individuals with severe nephritis, also high in individuals with double-stranded DNA autoantibodies, but it was much lower, as reflected by the lower odds ratio, in individuals who didn't have some of these other disease features. So there's quite a differential effect for STAT4 for different types of lupus. Similarly, a reanalysis of some of the recently completed genome-wide association study in European populations indicates that most of the recently identified risk factors for lupus are in fact relatively specific for double-stranded DNA positive disease. So here's that Manhattan plot in the double-stranded DNA positive subset with lots of great hits. In the double-stranded DNA negative subset, there's a little bit of an effect of HLA and IRF5, but not much else. And that's in spite of these being relatively equally powered in terms of patient number subsets. So still a whole lot more to learn about disease heterogeneity in specific genes. Now, lastly, there's been a lot of attention drawn to the fact that in spite of the many genes we've been able to identify over the last couple of years, we are still explaining only a portion of the heritability. And I've summarized some of this relationship between the number of loci identified, so about 25 for lupus, for example, with this estimate, and the percent of heritability explained. We're only explaining about 10% of the heritability in spite of all of these genes we've identified. Now, age-related macular degeneration looks much better in this respect, which was surprising. Most of the other diseases are characterized by this similar sort of pattern. So what explains the missing heritability? Maybe we need larger and larger studies to identify these variants with very small effects. Or maybe we need different studies, different marker sets, to identify rarer variants that have been left out of recent studies that have larger effects. Structural variants have also not been well captured by recent genetic studies, and they may explain a lot of the heritability in lupus, as may epigenetic modifications, which I'll describe in a little bit more detail in just a second. That is, modifications of the DNA that aren't DNA-based changes, but may explain heritability. Now, we have yet to perform systematically analyses of gene-gene and gene-environment interactions. Those may be important. But we certainly are dealing with a situation, at least in lupus, with imprecise phenotyping and disease heterogeneity that's interfering with our efforts. Now, epigenetics, this notion that there can be changes to your DNA that are inherited, that aren't due to underlying DNA-based changes, is very popular in a very broad audience. And there's been a lot of interest in looking at DNA methylation in lupus. You'll hear about that more later today and at this meeting. And I wanted to highlight one recent study that's pretty exciting and really implicates DNA methylation in lupus, perhaps more so than other autoimmune diseases. And in this study, the investigators took 5 identical twins discordant for lupus. Then they looked at methylation sites across the genome, and they compared them between the lupus twins and their healthy twins. And there was a striking difference between those identical twins, with the lupus twins having less DNA methylation overall compared to the non-lupus twins, and in particular for about 50 genes that turned out to have very important roles in immune response, cytokines, and cell activation. Now, what's shown here over on the right is those 5 twins with lupus with the hypomethylation pattern indicated by the blue, compared to their identical twins without lupus with the relative hypermethylation pattern, much more similar to a pattern in controls. So very intriguing data, and you're certainly going to be seeing a lot more studies like that in the future. So, tremendous progress since this last 8th International Congress 3 years ago, but still much more work remains to be done, replicating findings, extending to other populations to identify differences, identifying the causal gene variants, which for most of these recent discoveries, we are not at that point yet, refining genotype-phenotype associations, and finally understanding functional consequences of variants. But based on the recent progress and the insight already being provided by pathogenesis through these recent studies, hopefully this will contribute alongside the clinical trials and other efforts to help us identify better, more specific therapies and eventually better diagnostic and prognostic tools. Now, needless to say, this work is very expensive, involves tremendous amounts of expertise and resources, and there's been tremendous collaboration across institutions, individuals, consortia, some of which are listed on this slide. Many, many collaborators have contributed to these studies around the world. Some of the ones I've highlighted are listed on this slide. Of course, many hardworking research groups at the individual sites, such as my group at UCSF that I'm very proud about, and also most importantly, and I think I appreciate the opportunity at this session to highlight the important roles that patients have played in these studies. This is a family involved in our lupus study, and they just represents the hundreds and thousands of individuals who have so generously contributed their time to progress in this area. Thank you.
Should get a good description.
Lindsey's talk is open for questions. Please go to the microphones.
Lindsey, what struck me in looking at some of those Manhattan plots is that at least some of the so-called lupus susceptibility loci that were described, say, 10 years ago, before the GWAS era, are not well represented there. I'm thinking of chromosome 1q21 to 23, 1q41. What is your interpretation of the apparent discrepancy between what's being found in the GWAS studies and those loci that were so much focused on, say, 10 years ago?
Yeah, I think that part of what's going on is that for reasons of efficiency, we are utilizing standard sets of genetic markers for these studies. They're by no means comprehensive in terms of capturing even capturing common variation, let alone rare variants. So part of what's happening is that, we're more likely to pick up some previously identified genes and not others. And I think that explains at least part of what's going on. And also, I think, many of the genes that you may be thinking about are kind of buried in that borderline significance region. And possibly due to more modest effect sizes, they will reemerge as we pool together genome-wide association studies across populations.
Lindsey, that was very clear. I was wondering what you thought about the possibility that the susceptibility loci have odds ratios in the 1.5 to 2 range, so individually they don't seem to have much of a compelling story for an etiologic agent. But they could work in synergy, and thus in individuals, the effective odds ratio may be 10 or greater if the multiple loci are involved. versus an alternative hypothesis that each individual loci interacts with some environmental agent, and thus the combination of those are synergistic. And which do you think is a more likely scenario?
Well, I think several things are going on. First of all, the recently performed studies are just capturing common variants. We're probably underestimating the strength of association for at least some of these genes. When we look more carefully in specific regions at rarer variants, we will probably find some variants that may be more directly causally relevant with higher effect sizes. That's number one. Two, you're absolutely right. There's a lot of interest in looking at combinations of genes, especially within pathways, or combinations of genes and environmental factors. So far, and you'll hear about this subsequently at this meeting as well, we haven't been very successful at identifying significant gene-gene interactions. However, we shouldn't be so optimistic about doing that until we've really pinpointed the causal variant. So it may be a bit premature still to be looking at interaction effects. We will also need kind of novel methods that allow us to look for combinations across many, many genes than some of our traditional methods. And in terms of gene-environment interactions, one problem we have now is we don't understand well enough the specific environmental factors that are important to really specifically address those, although I think this area of DNA methylation may provide a nice example of gene-environment interactions. And so I think we may have a handle on some of that in the near future.
Thank you for a very nice talk. I would like to just ask you to comment on the issue that you raised in the beginning about the female predominance and how that suggested possible genetic components. And I'd like you to comment on your data and how your data now may provide some insight into that, or is that issue still a long way off from being resolved?
We still don't fully understand that, although there's some really intriguing work, for example, by investigators at OMRF that now, and other institutions, that implicate specific genes on the X chromosome. We know also that males that have the XXY karyotype are at increased risk of lupus, once again implicating genes on the X chromosome or some sort of a dose effect. I'm not sure that these studies can kind of shed light on that otherwise, although kind of hormone genes, hormone receptors have also been a focus of investigation. I'm not aware that there's been a lot of progress with respect to those sorts of genes through these recent studies, but Betty Sowes and others here are experts in that area and could address that for you, hopefully.
Hi, Lindsay. I'm Courtney Gray Montgomery. It's good to see you. Wonderful talk. Normally we just hear each other over conference calls, but good to see you in person. I just was going to make a comment and get your input as well. Certainly it's related to a comment that was made earlier about some of the discrepancies we see between the results of the GWAS studies and some of our earlier, typically linkage family-based studies. Certainly I'm of the mind, based on my training and history, that there are unique effects that are only going to be able to be identified in family studies, either because they are familial and not, sporadic or arising, out of population history, but also because they're recessive effects or they're very rare and that sort of thing. And I'd love to, and similarly for genetic gene-gene and gene-environment interactions, and I'd love to hear your input on that as well.
Yeah. First of all, we shouldn't expect to identify all the relevant genes through this sort of study design, case control. This is going to tell us about a lot of genes, not everything. I completely agree that there are probably, and to the extent this is true, we don't know yet, but there are probably some families, some individuals, some kind of specific, even individualized effects that are relevant to some patients or families with lupus that we're not going to see in these studies. But I think continuing to examine unusual families with lots of disease, early onset of disease, unique characteristics is probably going to be very informative as well. And we're certainly not in an era where we're putting those sorts of studies aside. They'll be complementary to these sorts of case control study designs, I think.
Thank you very much, Lindsay.